Well, we've made some ATP. That was a good thing. We've also made a whole bunch of NADH and FADH2s, and now we got to figure out how do we get the energy that we temporarily stored in these NADH and FADH2 molecules and get that energy converted so that we can make ATP. Well, that's what the electron transport chain is all about. The electron transport chain, or ETC, is basically another sequence or pathway of oxidation reduction reactions, just like the Krebs cycle was. The ultimate goal here is, of course, to make ATP. This series of events happens in the cristae of the mitochondrion. You remember you have the outer membrane of the mitochondrion, you have the inner membrane. On the inner membrane, there are a number of different enzymes which are involved in the electron transport chain. And they're housed in all of these different folds, these invaginations of the mitochondrion. Those invaginations are called the cristae. And what they do is they increase the total surface area inside the mitochondrion so I can have more duplicates of the enzymes needed for the electron transport chain, so I can have multiple electron transport chains functioning at the same time inside the mitochondrion. What's going to happen during the electron transport chain series of events is a process we call oxidative phosphorylation. And what that really means is we're going to consume oxygen, that's the oxidative part, and we're going to start putting phosphates onto 8 molecules of ADP. That's the phosphorylation part. Okay. So we're going to take inorganic phosphate and we're going to add it to adenosine diphosphate to make ATP. This is an endergonic reaction. We're going to have to use energy to get that phosphate molecule onto the ADP. And that energy is going to come from the stored energy in the NADH and the FADH2s from previously, and we're going to be consuming oxygen in the process of doing all these different steps. So let's take a look at this. Here's a artist's representation of the electron transport chain. And it involves a series of different enzymes, including something called NADHQ reductase. Here's one enzyme. Let's do the highlighter instead here. NADH2 reductase. Coenzyme Q, which is this little molecule here. We're going to get involved with another enzyme called cytochrome reductase. We're going to involve another shuttling protein called cytochrome C, and also another enzyme called cytochrome oxidase. And eventually we're going to start making actual molecules of ATP using a enzyme down here called ATP synthase. So in a nutshell, here's how this works. And by the way, there's a nice flow chart of this on figure 8.7 in your book that you might want to also check with. So remember, we are in the mitochondrion. Here's the matrix in kind of the dark pink color here. And you will remember that it is in the matrix that the Krebs cycle is occurring. And the Krebs cycle is making all these molecules of carbon dioxide, and it's making all this ATP, and it's making all this NADH and all this FADH2. So now we're going to take that NADH and FADH2 as it's being made. And remember again, those were temporary storage molecules for energy. And we're going to harvest that energy that's being temporarily stored in these coenzymes. So here we go. Here's a molecule of NADH. That NADH is going to interact with this protein right here called NADHQ reductase. And it's going to take the NADH and it's going to strip off the hydrogen. Okay? And that's going to cause basically that hydrogen to get moved over to the outside, out of the matrix, and into the fluid that surrounds the inner membrane here. Okay, so now we've got a hydrogen ion out here. And the electron that went with the hydrogen ion is going to get passed down to an enzyme called coenzyme Q. And coenzyme Q is like a little taxi cab. The electron gets onto coenzyme Q. Coenzyme Q physically shuttles or moves that electron over to the next enzyme called cytochrome reductase. And cytochrome reductase takes the electron it just got from NADH, and it also takes some electrons it gets from FADH2, and the electrons are it shuttles the electrons over to another enzyme called cytochrome C. It also takes the hydrogens that come off the FADH2 and moves those hydrogens out here into the fluid that surrounds the outside of the inner membrane. And that, by the way, this space here out here is called the inner membrane space. You'll probably remember that from your learning about mitochondria. Okay, so we're shuttling these hydrogen ions out into the inner membrane space. Meanwhile, we continue to pass the electrons on to cytochrome oxidase, and cytochrome oxidase is interesting. It's going to take those electrons and it's going to attach those electrons to an oxygen atom, and it's also going to grab a couple of hydrogen ions that are still floating around here in the matrix. It's going to combine them all together, an atom of oxygen, two atoms of hydrogen, and we're going to make a molecule of water, okay, H2O. And that is where we consume oxygen. Remember, the definition of cellular respiration is the consumption of oxygen and the production of carbon dioxide. Here's the step where we finally consume oxygen. It becomes the final electron acceptor of all of the events that we've gone through, including glycolysis, the Krebs cycle, the electron transport chain. The electrons are finally permanently accepted by oxygen. So oxygen is the final electron acceptor. Meanwhile, we're still building up lots and lots and lots of hydrogen ions out here in the inner membrane space, and that's important because we're building up a gradient here where the concentration of hydrogen ions is high in the inner membrane and relatively low in the matrix by comparison. And that sets up a nice scenario for something called chemiosmosis to occur. Chemiosmosis means the movement of atoms or molecules across a membrane. In this case, the movement of hydrogen ions across a membrane. Those hydrogen ions are going to move down their concentration gradient from where they're in high concentration in the inner membrane space to where they are in lower concentration in the matrix. And in the process of moving down their concentration gradient, they are going to be moving through an enzyme that helps to convert the energy there of movement to attach a phosphate ion to ADP to make a molecule of ATP. And this enzyme again is called ATP synthase. To synthesize means to make. So ATP synthase is an enzyme that makes ATP from ADP. And it does so by harvesting the energy of these hydrogen ions moving down their concentration gradient and back into the matrix. That's chemiosmosis. So then we make all of these molecules of ATP, and then we can shuttle the ATP molecules out of the mitochondrion, and they can be sent anywhere they are needed in the cell to perform whatever work is needed. So real quick, let's look at the whole process of oxidative respiration, this process of consuming oxygen and producing carbon dioxide. We started out with a molecule of glucose, and in the process of glycolysis, which occurred in the cytoplasm, we invested some ATP in the process, and we got some ATP out of the process as we split that glucose molecule apart into two molecules of pyruvate. Then we took those two molecules of pyruvate and we shuttled them into the matrix of the mitochondrion. And there we attached, we took one carbon group off of the pyruvate and attached some CoA to make molecules of acetyl-CoA. We then took the CoA off, and those acetyl groups entered into the citric acid cycle. I'm sorry, I misspoke there. We took the two molecules of pyruvate, we moved them into the matrix, attached the CoA to them, and in the process we got two carbon dioxide off. And now we have two acetyl molecules that enter into the citric acid cycle. Okay, now we have those two acetyl molecules entering into the citric acid cycle, and remember we involve a whole series of enzymes, and you should know well, at least kind of the order and idea here is we started out with citrate, we produced ketoglutarate, then we produced succinate, fumarate, and oxaloacetate. We got some ATP out of the deal by breaking apart bonds and releasing carbon dioxide. And we also made a bunch of NADH molecules and FADH2 molecules. And those NADH molecules that came from glycolysis and from the Krebs cycle are all going to be shuttled into the electron transport chain to make molecules of ATP. And it turns out the total number of ATP that we make is roughly about 36. Some books say 38, but it's 36 to 38 total molecules of ATP that we've made either through reactions during glycolysis, reactions during the Krebs cycle, or reactions during the electron transport chain. Pretty standard. Most people just say 36 ATP made, even though it could be as high as 38. Next up, we're going to talk about fermentation.